Hello, everyone. Thank you for joining this webinar this morning. The topic I'm going to share with you this morning is amino acids beyond body protein anabolism. There's emerging industry issues in the US. I'm going to introduce this issue first and then bring this to the aspect issues. And there was a reappearance of F18 E. coli outbreak in US commercial farm. And it has been a big issue in the US industry for a couple of months now. And in this case, this vet is actually found about 30% of mortality in worst case, or about the 60 to 75% of the morbidity in some better farms in here. And then he analyzed it, why that happens. And then, he suggested these 4 different, possible reasons for reappearance of the E. coli outbreak in the US industry. The first thing is the US industry started reducing or remove antibiotics. And then there was an increasing highly prolific sow introduction into breeding herd. And therefore, there was a compromised winner and whole herd immunity. And thirdly, there was an increasing divergence of genetic terminal structure duroc And that means that, if you use duroc lines in the breeding, the duroc lines basically have 4 times higher E. coli receptors in the gut. And therefore, they have more susceptible to E. coli outbreak in the commercial farm. And finally, there was an increasing other pathogenic bacterial population present in the farm, such as Salmonella, that actually increased the severity of E. coli outbreak. So if there is other pathogenic bacterial challenge, there's a more chance of E. coli outbreak can happen in the commercial farm. So emerging issues, all these 4 different causes are actually happening or going to happen in aspect market as well. Most of the aspect country is trying to reduce ADP use, and a lot of different aspect countries started introducing prolific sow genetics. And then I know that Vietnam actually having more than 30% of the prolific sow genetics now in the total industry herd. And also increasing, pathogenic bacterial challenge or presence of pathogenic bacteria in commercial farm in aspect countries is kind of a norm. So we have all these issues happening and going to happening in aspect, and these bacterial challenges or pathogenic bacterial outbreak it can be happen and can be a problem anytime sooner in our market. So what happens when that happens? If you look at this overview of immune system activation, when there's immune challenges, the immune cells such as monocytes, lymphocytes, or many other cells in the body start producing pro-inflammatory cytokines called interleukin-1 or TNF-alpha. And these 2 molecules are signaling molecules. They are sending signals to the liver and ask them to produce acute phase protein or other immune molecules using globulins in the liver. And then liver start using particular, selected amino acid to produce immune molecules or cytokines or, acute phase proteins. And if there is a shortage in amino acid in the bloodline, then they actually sending signals to skeletal muscles and ask them to, break down skeletal muscle and bring amino acid to the liver to respond to the immune system. They also send signals to fat and brains, and endocrine organs, and bone marrow, T cells. That whole signal is actually changing metabolic use of a nutrient. And those nutrients that actually used for this whole response is nutrient that otherwise should be utilized for body weight gain or protein deposition in the body. Therefore, animal performance is significantly reduced under immune system activation. So I'm going to introduce or focus this talk on functionality of amino acid in relation to intestinal inflammation. There are many other nutrients actually helping immune system response, but this talk is basically well concentrated on roles of amino acid in responding to intestinal inflammation. As you can see in this diagram, methionine is known to block the nuclear factor kappa B pathway, therefore reducing intestinal inflammation. And threonine also block nuclear factor kappa B signaling pathway, and also blocking mitogen activated protein kinase pathway, therefore reducing intestinal inflammation. Tryptophan has much diverse function, and they actually blocking MAPK pathway, and they actually increasing calcium sensing receptor expression, therefore reducing intestinal inflammation. And also, particularly tryptophan has ACE2 enzyme activity or increasing ACE2 enzyme activity, therefore reducing intestinal inflammation. So I'm going to concentrate on those three amino acids in today's talk. I'll start with the sulfur amino acid. If you need to understand involvement of the sulfur amino acid in the immune system, you have to understand methylation cycle. So we actually supplement methionine in the diet, and this methionine usually is utilized for normal body function or biological functions, or building muscles, so protein anabolism. But when animals get inflammation challenges, animals start using methionine and changing the methionine to S-adenosyl methionine. And this S-adenosyl methionine, called SAM, has an ability to donate one methyl group to DNA and histone for DNA methylation for gene expression. So when animals got challenged with inflammation or any other stress challenges, methionine being used to, as a methyl donors for DNA methylation, and DNA expression is actually being progressed to increase immune response, and therefore methionine is being used for this pathway. And then after donating, methyl group for DNA methylation, then this SAM is being converted to homocysteine, and this homocysteine in one way can be converted to cysteine and further converted to glutathione and taurine, which actually are being utilized to increase antioxidant capacity of the body. In other way, homocysteine can be converted back to methionine through the action or through the donation of methyl group from betaine or vitamin B12 or choline. And this, methionine resynthesized from homocysteine now can be utilized for protein synthesis in the body. So, at normal, healthy condition, methionine is used for protein synthesis, but in challenged condition, this methionine is being utilized for methyl donation, for DNA expression, and increasing antioxidant capacity in the body. Therefore, animals are in the methionine deficiency under such challenges. So sulfur amino acid is deficient when immune system is activated because it is being used for DNA and histone methylation, and a lot of different immune molecules contains high percentage of sulfur amino acid. Therefore, with immune system activation, sulfur amino acid are in short supply, and limited body protein deposition is happening after inflammatory challenges, and therefore animal sulfur amino acid requirement is increased under such conditions. And this is one of the group of finisher study conducted in Australia, and they had two different groups. The black line is healthy pigs, and red wine is the pigs that challenged with E. coli, lipopolysaccharide, E. coli, and the toxins. And they increased dietary sulfur amino acid from 0.45 to 0.75, and they measured protein deposition after 6 weeks of feeding challenges. And as you can see, when animals are healthy, increasing over the energy requirement of sulfur amino acid in the diet is not increasing protein deposition. But first thing, what happens when animals being challenged with endotoxins, you can see that there was a reduction in protein deposition, but by increasing amount of the sulfur amino acid in the diet, then there was an increase in protein deposition because we're actually supplementing those methionine and cysteine that is actually utilized for response to immune system activation, and therefore a shortage are supplemented for muscle growth, and therefore protein deposition is being increased by supplementation of sulfur amino acid over current requirement of methionine and cysteine. And very similar study conducted in Netherlands using wiener pigs. At this time, they used E. coli challenges through the oral route, and they measured sulfur amino acid requirement in response to in terms of body weight gain, and also in terms of a gain to feed ratio in right hand side. As you can see in the left hand side, this is the energy requirement of sulfur amino acid. And you see that it's not really supporting optimal growth on the E. coli challenges. And on the E. coli challenge, the optimum growth rate was achieved at about 20% higher or 25% higher sulfur amino acid content in the diet than current energy recommendation. And this very similar response for gain to feed ratio as well. And this wiener pig study basically shows very similar response that I presented in Grow Finisher study as well. And this study is pretty interesting study that was published in 2018 in on the Livestock and Feed Business magazine. And in this case, they actually had 2 dietary challenges. First one with AGP. At this time, they used the 1% of macros 2.5. And the other diet didn't contain any AGP. So they removed the AGP in orange colors treatment. And they increased dietary sulfur amino acid content in the diet. And what they found, very similar response, that when they had AGP in the diet, increasing sulfur amino acid didn't really improve FCR. But when they removed AGP in the diet, the first thing they see is deteriorating FCR, in the lower sulfur amino acid contained diet. But by increasing sulfur amino acid in the diet, they improved hormones or FCR response, even without FCR, without antibiotic inclusion in the diet. That was the end of the sulfur amino acid story. Now I'm moving on to tryptophan. Tryptophan is degraded to its metabolite, first metabolite, chyminin, and secondary metabolite, as you can see in here. And they are free radical scavengers. And this degradation of tryptophan happens when there's increase in inflammatory response by increasing pro-inflammatory cytokines such as interleukin-1, interleukin-6, and TNF-alpha, or if there is an increasing stress response by increasing glucocorticoids such as cortisol, then these two, stimulation or inflammatory stimulation or stress stimulation is basically stimulating 2 different enzymes called indolamine 2,3 dioxygenase or tryptophan dioxygenase that actually stimulating degradation of tryptophan to its metabolites. And tryptophan metabolites basically stimulating enterocyte epithelial cells to increase production of antimicrobial peptides. And these antimicrobial peptides produced in the epithelial cells are secreted into the surface of the gut. Therefore, they are protecting gut from further inflammation from pathogenic bacteria. Because of that, this Winnipeg study conducted in Italy, they found that there was about 17% to 18% increase in average daily gain and average daily feed intake if we increase, tryptophan to lysine ratio from current 17% recommendation to 21% recommendation. So I know that a lot of industry actually implementing higher levels of tryptophan than NRC recommendation because of this, this functionality. Particularly in the U.S. industry, they basically, supplementing around the 19 to 20% tryptophan in the diet. And this is old study conducted in France. And they used this time health challenges, lung infection challenges, and altered, tryptophan content in the diet from 15% to 20% in relation to lysine. They measured body weight gains. And as you can see in here, in a healthy condition, when you supplement NRC levels of tryptophan in the diet, animals actually reaching optimal body weight gain or optimal growth weight. When animals health challenged or lung infection challenged, higher tryptophan content is optimizing body weight gain in piglets. Therefore, higher supplementation of tryptophan under such condition will benefit animals' growth weight or overall herd growth performance. And this is one of the Australian study conducted in Winnipeg after infection with E. coli. And they measured APP index. APP index is measuring 3 different acute phase proteins and expressing as an index. And you can see that APP index in here can be considered as an inflammatory response in pigs. So higher the APP index means a higher inflammatory response. And they actually measured this APP index before infection, day 5, and 25-hour infection at day 8 to see the short-term response. And then day 14 after infection, which is 7 days after infection, to see the long-term response. And as you can see in here, low tryptophan and low sulfur amino acid represents current NRC levels of tryptophan and sulfur amino acid. You see that inflammatory response increased after infection, and that is actually lasted for more than 7 days. But when you supplementing higher tryptophan levels, then there was a reduction in short-term inflammatory response, but it is not really affecting the long-term inflammatory response. So tryptophan, when you're increasing tryptophan in the diet, is really helping short-term response to the inflammation, but not really to the long-term response. But when you're increasing sulfur amino acid, it basically reduces short-term and long-term inflammatory response. And when you're supplementing tryptophan and sulfur amino acid together, you see that inflammatory response will, suppress much better compared to just supplementing tryptophan and sulfur amino acid to current NRC recommendation. And from the same study, as you can see that performance was maximized by supplementing both sulfur amino acid and tryptophan rather than individual supplementation of the sulfur amino acid and tryptophan, and also for FCR response as well. Now I'm going to introduce a new concept of dietary formulation, and this time I want to discuss about tryptophan to large neutral amino acid ratio in the diet. Large neutral amino acid is includes isoleucine, leucine, valine, phenylalanine, and tyrosin in this case. We feed tryptophan in the diet, and about 10% of the dietary tryptophan pass across the blood-brain barrier and enter into the brain. And this tryptophan converted to 5-hydroxytryptophan, and this 5-hydroxytryptophan converted to 5-hydroxytryptamine in here. This 5-hydroxytryptamine is serotonin, which is very well-known hormone that sending negative signals to HP axis of stress axis. So hypothalamus, pituitary, and adrenal gland axis, and then therefore they're actually reducing stress hormone production. If you look at this blood-brain barrier, which is very, very, having very strange arrangement in here, but it is understandable. So if you look at this barrier, in the brain side, they have all different sort of sodium-dependent amino acid transporters. The reason being is they want to get rid of any excess amino acid in the brain to the outside of the brain. But if you look at this blood side, there's only 2 amino acid transporters, and this leucine preferring transporter is most functional amino acid transporter in the blood side, which means the brain doesn't want to, too much amino acid being transported into the brain. But the fact is, tryptophan and large neutral amino acids using these same leucine preferring transporters to get into the brain. Therefore, they compete each other to get into the brain. So if you have too much of large neutral amino acid or too small amount of tryptophan in your diet, then amount of tryptophan going into the brain will be reduced, and therefore animals will be more prone to stress. So basically, tryptophan compared with large neutral amino acid to cross blood-brain barrier. And there's more evidence, supplemented or conducted with the lab study. And as you can see in here, if there's an increasing serum tryptophan content, and there's a linear increase in cortex tryptophan, and then increasing cortex tryptophan content, linearly increase hypothalamic serotonin concentration. And it's been proved in piglet study in the US by increasing tryptophan content in the diet from 0.17 to 0.33% of the lysine. There was a linear increase in plasma serotonin content, and there was a linear reduction in salivary cortisol content. So higher the tryptophan content in diet, animals' stress response will be much, much better and reduced. And in Australia, we conducted one piglet study with the E. coli infection model, and we had this time 4 different diets. So current NRC levels of tryptophan and high large neutral amino acid in the diet, which is basically normal in corn soy-based diet. And then second diet, we actually reduced some of the large neutral amino acid. And third diet, increased tryptophan. And fourth diet, both increased tryptophan and reduced large neutral amino acid. By doing so, we actually changed tryptophan to large neutral amino acid ratio from 0.044 to increase 0.074%. And what we found in the blood measurement, we found that there was a very strong trend in increasing serum serotonin content in diet 4, which is diet increased tryptophan levels and also decreased large neutral amino acid. But in the diet, just increased tryptophan or just decreased large neutral amino acid didn't really improve every daily gain and serum increase any serotonin content. So in the commercial levels of dietary formulation, if you can increase some of the tryptophan or limit some of the large natural amino acid, then your animals will be much more, stronger for any social or environmental stress in the commercial farm. Okay, now I'm moving on to the threonine this time. Threonine is the major amino acid for mucin and immunoglobulins as well. So in this case, in layer study, they had two different control diet with a 16% crude protein, and reduced protein diet at 14% crude protein. And then they actually had reduced protein diet with about 0.3% of the threonine included in the diet. And as you can see here, including more threonine increased the MOC2 gene expression, which is mucus producing gene. And also there was a very significant increase in secretory immunoglobulin G messenger RNA expression in the mucosal cells. So higher the threonine, you will expect better protection of the gut by producing more mucin, and better protection by increasing antibody production in the mucosal level. And because of that, this study actually had a very similar setup with the previous study, that they included AGP in the control diet with 30 ppm of selenomycin, and another diet, that diet didn't actually had selenomycin included in the diet, so Adp Free Diet. And they measured performance response from 25 to 110 kilograms. And as you can see in here, very similar response from other amino acid study, that if you include AGP, changing threonine levels in the diet didn't really change any performance response. But when you removing antibiotics in your diet, the first thing you see is reduced performance, reduced growth rate, increased FCR in the right-hand side, you can see. But by increasing threonine content in the diet, you can see that there was an increase in everyday daily gain and reduction in FCR. And this study particularly used combination of 3 amino acids. So they had two different sanitary challenges. Sanitary challenge, like open bar indicates a low sanitary condition, so animals are exposed to dirty condition, and the closed bar indicates that animals in high sanitary condition, like a very clean and research environment condition. Then they fed two different diets. Base amino acid diet, which just contained enhanced levels of the amino acid. And then amino acid supplement diet contained 20% higher methionine, threonine, tryptophan compared to enhanced requirement. Then they fed the diet, and they found significant interaction, which is in line with all the other amino acid in antibiotic studies, that in the animals in the high sanitary condition, there was no differences or no change in white blood cell count and gain to feed ratio by supplementing more amino acid. But when animals are exposed to dirty condition or low sanitary condition, there was a significant increase in white blood cell count in plasma, and also gain to feed ratio for growth initial pigs. Another study looked at the functional amino acid in Salmonella challenged pigs, which was a very recent study conducted in Canada. And they found that by supplementing 20% higher the requirement of methionine, threonine, and tryptophan, AA plus in here, reduced and significantly reduced Salmonella excretion in the feces after inoculation compared to control diet. And then they found also in the control, which is not challenged animals, and ST, which is Salmonella tripurium challenged animals. You can see that there was a significant increase in fecal MPO expression. MPO is a peroxidase enzyme that expressed in neutrophil granulocytes. So higher the MPO means more of intestinal damage. So that is the indicator of intestinal damage used. And as you can see in here, by supplementing functional amino acid, intestinal damage index is significantly reduced in the Salmonella challenged animals. And they also measured post-inoculation performance for 7 days after Salmonella challenges. And then they tested in the low protein and high protein diet. And as you can see in here, there was a very significant increase in everyday gain, regardless of protein content in diet. And there was a very significant improvement in FCR by supplementing functional amino acid in Salmonella infected pigs. And this is another very, very interesting study. And this time, this Canadian group actually asked a question that then how long should we feed this functional amino acid supplemented diet to reduce inflammatory response in Salmonella challenged pigs? And then they had 4 different treatments. FAA minus is just control diet without any amino acid supplementation. And FAA plus 0 is amino acid supplement at 20% higher levels of methionine, tryptophan, and threonine. But they fed this supplement diet at from the day that they infected Salmonella. And then FAA plus 1 means they fed this functional amino acid supplement diet 1 week before infection. And FAA 2 means they fed this functional amino acid supplement diet 2 weeks before inflammation. Then they found that, very interestingly, Salmonella count in colonic content was linearly decreased by feeding this functional amino acid supplemented diet 2 weeks earlier than Salmonella inflammation. So basically, if you supplement higher amount of this functional amino acid in your diet and feed them to commercial farm animals, and if there was a E. coli or Salmonella challenge or 2 weeks and 3 weeks after the start of this diet, feeding this diet, then those animal group will have a much, much better response to inflammatory challenges. And this is very interesting piece of information. They also measured serum albumin and serum heptoglobin levels when animals got challenged or inflammation challenge or stress challenge. The albumin content in the blood is reduced and heptoglobin content is increased. But by feeding functional amino acid 1 or 2 weeks earlier to challenges, you can see that there was an increase in albumin content and decrease in heptoglobin content, which is very similar response to Salmonella count in colonic content. Therefore, animals are responding much better to Salmonella challenges with feeding this functional amino acid supplement diet 2 weeks earlier. And you can see this post-inoculation performance gain, feeding functional amino acid supplement diet earlier than actual pathogenic bacterial inflammation challenge or inflammation is animals' performance was much better compared to feeding that diet from after the event of the inflammation. Another study conducted in this Canadian group is measuring intestinal alkaline phosphatase expression in the ileum. Intestinal alkaline phosphatase is a brush-border enzyme that detoxificates microbial endotoxins and therefore inhibit intestinal inflammation. And if you increase expression of intestinal alkaline phosphatase, then intestinal inflammation is generally downregulated. So in this study, in the first study, they actually fed two different diets, with and without functional amino acid supplement diet, 20% higher methionine, threonine, and tryptophan in low protein and high protein diet. But regardless of protein content, ileal alkaline phosphatase expression was increased in the piglet that actually supplemented with 20% higher functional amino acid. Now they also had a look at how long this functional amino acid supplemented diet should be fed if you want to increase intestinal alkaline phosphatase expression. And it is very clear to show that any time from inflammation, if you increase functional amino acid content in the diet, ileal alkaline phosphatase expression will be increased. So that is the end of my first part of the presentation that actually showed functionality of these three major amino acids. And then, in the next section that I'm going to introduce what is happening in our CJ research station, that we're actually trying to enhance functionality of this specific amino acid through, changing the forms, like a granulation of amino acid with Corynebacterium protein, and also probiotics included in amino acid. So in the first place, we actually measured anti-inflammatory capacity in here. In this case, we measured interleukin-8 reduction capacity that was actually measured in the intestinal porcine enterocyte cell line, jejunum cell line. We actually treated with the different amino acid, and challenged the cell with mycotoxin DON. And then we measured pro-inflammatory cytokine interleukin-8 production, and expressed in here how interleukin-8 production was reduced in different amino acid treated groups. And as you can see in here, in the methionine group, particularly in here, compared to DL-methionine, L-methionine, and L-methionine Pro, which contains about 5 or 10% of Corynebacterium postbiotics, had much higher interleukin-8 reduction capacity, which indicates that anti-inflammatory capacity can be enhanced by using L-form methionine compared to DL-form methionine. And we also measured interleukin-10 content in the porcine bone marrow-derived macrophage, which is immune cells. And then we treated with amino acid, and we measured interleukin-10 production as anti-inflammatory cytokine production ability. And as you can see in here, again, L-methionine, and particularly L-methionine Pro, which contains about 10% of Corynebacterium postbiotics metabolites, increased anti-inflammatory cytokine production in macrophages. And if you look at tryptophan in here, again, tryptophan Pro, which contains Corynebacterium postbiotics, showed very much improved interleukin-10 production capacity. So antioxidant capacity or anti-inflammatory capacity basically improved with L-form methionine, and with supplementation of 5 to 10% of postbiotics in the amino acid. Then we measured antioxidant capacity by using the intestinal cell line, amino acid treated with hydrogen peroxide. And then we measured reactive oxygen space production, and we expressed in here as a reactive oxygen spaces reduction rate in here. And as you can see in here, if you look at the methionine compared to the L-methionine, L-methionine is increased, reactive oxygen space reduction, so antioxidant capacity, and also tryptophan, particularly tryptophan Pro, and then also threonine Pro, and CJ Bio L-threonine had much better antioxidant capacity compared to competitor products. And finally, we had to look at the anti-heat stress capacity in here. So in this case, we used intestinal cell line and macrophage in the bone marrow cell line. Amino acid was treated at 42 degrees for 2 hours, which is inducing heat stress. And we measured heat shock protein production, and then we expressed heat shock protein 70 production rate in here. So as you can see in here, compared to the L-methionine, again, L-methionine and L-methionine Pro, and particularly tryptophan Pro, increased heat shock protein 70 production, which means they had a much better anti-heat stress capacity. And also in the immune cells, we found that very similar response that L-methionine, L-methionine Pro, and tryptophan, particularly tryptophan Pro, actually increase anti-heat stress capacity of amino acid. And then overall, if we summarize these studies, in case of methionine, by using L form over DL form, can increase antioxidant capacity, anti-inflammatory capacity, and some anti-heat stress capacity. And also threonine, by using L-threonine Pro, which contains some of the Corynebacterium metabolite, can increase antioxidation capacity, and tryptophan as well by supplementing some of the Corynebacterium cell content and postbiotics, we could increase, particularly anti-heat stress capacity and cell growth and anti-inflammation capacity. So that was all of my presentation today. If I conclude this morning's session, under environmental and inflammatory challenges, monogastric animals preferentially use selected amino acids for maintenance of biological function, particularly immune functions. Therefore, these amino acids will be periodically deficient and possibly limit optimal protein deposition and therefore reduce growth rate. And then recent research data in the literature very well demonstrate that additional supplementation of methionine, tryptophan, threonine at around 20% of the requirement can improve resilience to inflammation challenges, improve epithelial defense mechanism, and improve growth performance. And at CJ Bio, we are further developing amino acid functionality by granulating amino acid with Corynebacterium protein and Corynebacterium postbiotic, which showed very promising result as we presented in here. So that is all my presentation, and thank you for your attention. And I'll pass stage to my colleagues here. Thank you.